Thank you, Doug, for that introduction and for hiring me. It's been a wonderful 2 years. Um, President Guskowitz and members of the Board of Trustees and members of the MSU community, I'm really excited to share with you our work on plant resilience research. So I'm going to start by introducing Plant Resilience Institute, or PRI. Doug has already mentioned that this was established 10 years ago by the Global Impact Initiative. Today, we have 14 faculty across colleges and departments, and we're engaged in interdisciplinary, very collaborative, and cross-scale plant science research, looking at plants all the way from atomic and molecular levels to ecosystem levels. The crazy graph on the right is a circus plot that shows all the lines of collaborations that we have among PRI labs. So today, PRI is ranked at the very top of about 40 plant science departments and training programs in the US. The vertical axis shows you the size of the units, and the x-axis is the rank. So PRI shares this highest rank with 3 other institutes, but as you can see, it's the smallest. And so with a fairly small and highly collaborative and successful team of scientists, what can we aspire to do? So in the next 10 years, this is what we'd like to do. First, we would like to be known for establishing field to lab to field paradigm. So today, a lot of fundamental research is done in the lab, and the discoveries are taken to the field to translate, but often this does not work. In fact, most of the times it doesn't work. So we'd like to turn this around and do fundamental research in the field and take the findings back to the lab, find out the mechanisms, and take that back to the field. We'd also like to integrate fundamental science with translational and applied science in plants. And I think that MSU is uniquely positioned to do this very well. Finally, we'd like to do our research using team science approaches at the global scale. So recently, some of these aspirations have manifested into a project that I'd like to talk to you about a little bit more. This project, as Doug mentioned, is called Center for Sustainable Plant Innovation and Resilience through International Teamwork. It's a little bit of a mouthful, so we call it Sea-Spirit. Sea-Spirit's aims are to discover natural products from plants and microbes to improve plant health. This is a collaboration with 5 countries, US, Japan, UK, Canada, and South Korea, and we got funding from the 5 respective countries' funding agencies. We're gonna be working together on 7 crops that are listed here. These are globally traded commodity crops like corn, soybean, and rice, and so on. And then we're gonna target some of the worst stresses that we're facing today, such as drought, heat, salinity, and fungal and nematode pests. So how are we going to try to identify these novel bioactive compounds? We have 2 strategies. One is to expose plants to stress like heat and drought, and we're going to extract the compounds and see if we can find compounds that are induced by stress and perhaps provide protective roles for the plants. A second strategy we're taking is to develop high throughput screens to find these bioactive compounds by screening natural product chemical libraries, basically using drug discovery pipelines. Okay, so I'm just going to tell you we're doing a lot of different studies and experiments in this project so far, even though it just started a year ago. I'm just going to tell you about one study that we did this summer at Macomb Field Station. In this study, we asked how do heat-tolerant and heat-sensitive potato varieties respond to heat in the field, okay? And so we went out to the field every 2 weeks and studied heat-sensitive and heat-tolerant potato varieties and provided this heat for 2 weeks at a time during potato tuber development. And we used these tents of heating arrays. And then we also took photosynthesis measurements and then took leaf samples back to the lab to do molecular work. This was a really extensive collaboration among scientists in extension, biochemistry, engineering, and mass spec facility. And so we have a lot of interesting data, but today I'm going to just show you one result. Okay, and this is stomatal conductance, which is basically how plants breathe, take in carbon dioxide, and release water and how plants breathe and sweat, really. And so as you can see in this figure, the heat-sensitive variety increased its stomatal conductance in response to heat. You can see the orange dots in the box there, but the heat-resistant or tolerant variety didn't budge. It didn't change the stomatal conductance. So what does this mean? Are the sensitive plants trying to sweat and cool off more? We don't know. So we're going to try to understand what's going on by actually conducting these experiments now back in the greenhouse and some growth chambers. So this is led by an amazing postdoc, Kathy Rivera Zuluaga, along with a couple of visiting scholars from Japan, Shogo and Kwangchul. And we're using this very nice automated watering system that Berkeley Walker from PRL built. And of course, this very beautiful, beautifully renovated greenhouses and head house. Thank you so much. That Chrislyn Partica is running. In addition, we're mimicking the field summer conditions in our growth chambers. And again, we're really lucky to have someone like Cody Kylan, who's running the growth chamber facility. And we have extremely nice chambers that can really replicate the field conditions right in our labs. And I want to also thank Corinne Prado, who is a lab manager in my group, who's doing a lot of these operations. So the second strategy that we're taking is really leveraging MSU's drug discovery core. And what we want to do is to develop these high throughput screening assays using chemical libraries to find new antifungals and new protective compounds from plants. The fungus that we're going to start with is Fusarium. Fusarium species reduce billions of dollars of crop loss every year globally. And these are some of the diseases that are caused by Fusarium in potato, tomato, soybean, and maize. So we're partnering with Erica Elizabeth at MSU Drug Discovery Core to develop a high throughput fungal assays to find new fungicides, basically. And so here we're using two species of Fusarium, Oxysporum and Vigiliforme. These are kindly provided by our pathology colleagues, Linda Hansen and Marty Chilvers. Again, this project is led by Kathy. So because I'm not a pathologist, I did not know that there are only 3 major classes of commercially available fungicides used in agriculture today. And you can see the classes look kind of similar in structure, and they all target only one pathway, and that's ergosterol. Ergosterol is sterol, found only in fungi, okay? It's very important for membrane function, and you can imagine that there may be some pathogenic fungi out there that are already building resistance against these drugs that target only one pathway. In fact, Kathy found that one of the commercially available fungicides, fludioxanil, actually is in fact not effective on Oxysporum. So the left graph shows you control that has no fungus. So you can see there's no growth, all the lines in the bottom. Oxysporum, doesn't matter how much of this fungicide we provide, we always see growth. So this fungus is actually resistant to fludioxanil. However, Vigiliforme is actually sensitive. We can kill it at a low dosage, as low as 1 parts per million of this fungicide. That plate that I'm showing you has 384 wells, and each well allows us to do an assay. So we're going to do high throughput screening of about 30,000 chemical libraries and ask questions like, what stage of the fungal growth are these compounds acting, and what concentration of these compounds can we kill half or even 90% of the fungus? Once we find these hits, we're going to immediately go test to see if they're toxic in the environment and on animals, in collaboration with Carrie San, who's a toxicologist here at MSU. And then we're going to test these compounds on plants and then eventually go into the field. So I want to end by saying that at PRI, we are developing a framework to accelerate discoveries and solutions for future-proofing our world through resilient plants and plant resilience research. And we're doing this by establishing field to lab to field paradigm. I gave you an example of that already. And we are also applying other approaches that are described here, but we won't have time to talk to today, but it's actually described in an article in Transcend Plant Science that was published last year. We have over 110 members of Seaspirit today, so I can't name them all, but I wanted to share with you some photos to show that we're really having a great time, not only great science, but people are really getting to know each other across these 5 countries. And I have to thank the people that I work with every day and bring me a lot of joy. These are members of my lab and the PRI admin team who come from all over the world and hopefully will go back and make more impact. Thank you so much for your attention.
Any questions for Dr. Rhee? I have one, or two maybe. One is that I know as we celebrated at the university speech that we gave a few weeks ago, that apples, we know that we're the second leading apple producer Michigan is. You mentioned potatoes. I'm curious as to where we sit in terms of potato production.
25% of potato chip potatoes come from
Wow.
Michigan.
Okay. Wow.
And the 2 varieties that I told you about, I'm not going to reveal the names, but they are used in potato chip making.
Wonderful.
Today. Yeah, they're commercial varieties.
And then just as we learned when we were up near Traverse City for our board retreat in June, we had a chance to visit a vineyard, and they talked about how climate change is affecting the production of grapes and wine. How is it affecting, how does your research intersect with the climate change and creating more resiliency for crop growing?
Yeah, thank you for that. Actually, fungal pathogens are the #1 pests of all of the types of pathogens around the world, including grapes. So it's actually quite a devastating type of infection because they affect not only the quantity of yield, but also they produce these very bad toxins called mycotoxins that make the grapes or corn grains unusable. So because they affect animals and humans. And so it is really a bad problem. And the fact that we're starting out with finding novel fungicides, I think is very meaningful. And the fact that there are actually not a lot of varieties of fungicides that are used commercially and that they're all synthetic ones. The ones that we're trying to develop are natural products and like hopefully biodegradable and environmentally safe. And, we may not be able to patent them because if they're already like well-known or FDA approved already, but we care more about farmers being able to get access to these compounds right away. They could maybe go to CVS and then using them that season. So, I didn't mention this, but the traditional way of trying to improve agriculture is breeding and these more recently engineering, but these processes take decades. I think that if we're able to find some safe, environmentally safe and animal safe compounds that farmers can use like right away, we can help them next year. And maybe temporary solution for that season that has like a major heat wave or drought spell, but we can still like help the farmers that year, not, lose 80% of their crop.
I have a quick question if I can. So one of the first things when, I guess, 6 years ago now, Chair Tebay and Vice Chair Scott and I saw were the greenhouses that did not look like the photo that we saw here. And it took a while. It took a number of years. I think all of us visited at least twice in their former form and saw just how much you needed that investment and that improvement. And I really appreciate that you mentioned it now, but I wonder if you would just say a little bit more about what it has meant for the university to be able to invest in that infrastructure to support your research.
Yeah, I mean, these greenhouses are really life-changing. I mean, growth chambers too. Before I came here, we had to custom build these greenhouses and growth chambers to mimic conditions of the fields. And we are very interested in mimicking conditions of the future. And, Corinne Prado that I mentioned, she literally babysat the growth chambers like 12 hours a day, okay? She came here, we got these amazing chambers. Thank you, Doug, and thank you, everyone. She's literally told me, I quote, "This was life-changing," and because now she can actually do real work. Same thing with the greenhouses. We had geysers in the middle of the greenhouses before, you probably saw that. And now, we can control the, conditions and, they work beautifully and we have space and there's a lot more collaboration that can happen. the automated watering system that I told you about, I don't think we could have put that into the old greenhouses because why would you want to set up a fancy automation when the rest of the condition is like not quite ready? So yeah, it's really allowing us to think about, okay, now we got really controllable conditions in the greenhouses. What else can we do? Like, how can we push ourselves and our science? Like now we're actually thinking about automation. We're talking about like digital twins. We're going to get into AI. High throughput phenotyping, we can start thinking about these things. So I think that what you can do, we just basically, on top of that, synergize, and it just kind of goes into kind of exponential mode of discovery, which is really exciting.
Great.
Thank you.
Oh,
Uh,
I'll go. No. It's Dr. Gage, I just want to thank you. Every time you bring someone, we learn something, and I like to learn, so thank you. I didn't know what pennycress was. I had to look it up. Yeah, I looked it up.
Yeah.
So you're using it it's not edible, because I like food, and I had never heard of pennycress.
I'm so glad you mentioned that because I actually neglected to bring that up. It's an emerging cover crop. It used to be a noxious weed only about 5 years ago, but plant scientists have actually domesticated it using genetic engineering. And it's an oilseed crop that's winter hardy. So you can plant it in between your corn and soybean rotations. And so after you harvest your corn or soybean, you plant pennycress, and over winter it grows. And in early spring, farmers can harvest the seeds for oil. It makes like really nice oil for sustainable jet fuel. And it also covers the ground so that when it rains over winter, you don't have soil erosion and you don't have nutrient bleaching. So it's called like a cash crop that's cover crop is called intermediate crop. So pennycress is one of the exceptions of the 7 crops that we're doing. It's not a world globally traded commodity. It's a cover crop, but we're really excited about pennycress. In fact, we just planted our first, we started doing our first field study here at MSU this fall. We just planted the seeds in September. So actually I have a picture. This is it. Actually, I wanted to show you this, but I didn't think I had time. So in 2 weeks of like learning that we had some space available, we went to Mike Partika, who's the agronomy farm manager, and 2 weeks later, we were able to plant the seeds. This is my postdoc, Danielle, Paige Smith, who's a PRI germplasm curator, an amazing grad student, Brianna Hashem, in biochemistry. The 4 of us just actually went out to the field, checked it out. 2 weeks later, we had seeds on the ground. So, yeah.
Okay, so don't eat.
Yeah, no, you can eat the
So-
oil after extraction, but no, it's not. No.
Yeah,
But
I love food, and
But it is similar to arugula, so you could try. if you
I'm not
want.
gonna try it, but thank you. Thank you, though. I appreciate the invitation. So the other thing you mentioned soy and corn, and I was looking at your afflictions, and they were like sudden death syndrome for soy and ear rot. Are those real, or did you make those up?
They are real, and they are epidemic.
I mean, the names.
I didn't make it up, but it's
Mhm.
that sudden death syndrome is actually one of the worst epidemics we're facing today in the U.S.
Wow,
Yeah.
for soy.
for soybean. Yes.
Wow. And then my last question was back to that map. Having visited Africa maybe 12, 13 times, why and literally agriculture is everywhere. It's
Yeah.
like a mashup
Yeah.
of industry and industrial and
Yeah.
agriculture. Like you walk down the street and there's goats with you,
Yes.
right? But it's like in the middle of Addis Ababa, like
Yeah.
in real life. Why are you not there? What's happening?
Yes, I'm glad you mentioned that because when I was making this map, I saw it too. And I thought about like, oh, is it cool to highlight this? And I thought, yeah, it is because I really want to go there. And I want to also go South America and Australia, the areas that are not covered. So it tells me that, okay, we are very diverse in our group, but not diverse enough. So I'm going to work on that.
Well, if you need help in Ghana, Ethiopia, Nigeria, South
Yes.
Africa, call me.
I will. I'm going to. I'll email you.
Thank you.
Thank you.
Thank you.
Provost McIntyre.
Thank you, Professor Rhee. This is really incredible. I wonder if you could talk a little bit more, kind of piggybacking on what Trustee Vassar was saying about the importance of the global work to accelerating the impact of your discovery. So you're working across 5 different countries. Talk a little bit about what that means and what those global partnerships mean to MSU.
Yeah, for me, it's been really an eye-opening experience. I thought that it would be very, very challenging because of the differences in cultures and how we operate in science. But in fact, I think these differences actually have become such a great opportunities and learning lessons. And we have different problems that we face in agriculture, in our economy, in policy. And so it's really enriching us to sort of be more, I guess, open-minded and to look outwards. And I think our trainees are loving it. Okay, so we have already trained 4 scholars from Japan so far, and we have a constant flow of people coming. I'd like to actually try to do the reverse of sending our trainees to other countries as well through this project. But yeah, it's really been a huge learning experience, and I am like kind of swearing by it now. I think we should really be more and more global.
I just have one question, thanks. Thank you, Dr. Rhee, that was fascinating. If you look at the chart that you put up of all the plant science programs in the United States, as well as what you're doing with Sea Spirit, how many are using the field to lab to field, and how many are using AI technology?
A lot of people are trying to use AI. The field to lab to field is new. It's something that we're trying to do. There's a scientist, Andrew Leakey at University of Illinois at Urbana-Champaign, who has sort of done something like field to lab to field, but not in the way that we're envisioning, but he's actually more advanced than we are. He's got this amazing field research infrastructure. It's like massive green rain out shelter that's from production, like companies that make these devices for farmers that do sensitive vegetables and cut flowers and things like that. He's got like a 1/4 of an acre size of these rain out shelters that can actually close and open within 2 minutes of rain. So you can really precisely control environments. And so we want to replicate that here at MSU, and we're trying very hard. And we would ultimately like to connect and maybe have these field research stations that are doing fundamental research. And I don't know if you know, but fundamental and translational research. NIH has funded 60 translational centers for medical research. I think they're called CTSAs. And for plants, there is 0. We have no translational research centers out in the fields, which is our equivalent to clinical trials. So we are really excited to establish that, especially in the Midwest. I just recently learned that the Great Lakes megalopolis is the biggest region in North America with 60 million people. The amount of, it's second in terms of GDP to the Boston, Washington area. But its investment into this area is, of course, much, much, much lower than the Boston, DC area. And it's because that area gets a lot of money for medical research, right? I think we have an opportunity to bring in that investment for agriculture and really help the entire region, which has got the most number of people.
Trustee Balo, did you have a question? You don't? Okay, great. Well, thank you so much. I'm just thrilled. I love potato chips. I'm just glad we're not going to run out here in Michigan anytime soon. And great presentation. Appreciate all that you're doing.
Thank you,
Thank you.
President Esquith.